I'm the one who's introducing today Nils. Okay, I understand that. So Nils Volkenborn is associate professor at the School of Marine and Atmospheric Sciences at Stony Brook University. So Nils is interested in the functioning of the seafloor, specifically in the relation between benthic species and sediment biogeochemistry. Nils is used to use experimental approaching from large-scale field and manipulation to laboratory studies on the scale of individual organisms. He is also a tech guy, developing and using micro-scale sensors technology and imaging techniques. He's already in love with Arenicola marina, and he tried to stage a coup in order to turn the name of the Nereis park into an Arenicola park. But okay, you can pass this one. So he will now present us some result about an insight about different works he did with its favorite species, Arenicola. Thanks, Nils, for doing that for us today.
Well, thank you very much for inviting me for this seminar. And yeah, it's a pleasure to be here and see on the participant list several familiar names, some new names. And so I'm excited to present data today from actually throughout my scientific career, because when I was asked or invited to give a seminar, I was thinking, okay, what should I present on? And I decided actually this is a good time point to maybe look back to what I have done with many collaborators over the last 20 years now, since I started a large-scale lugworm exclusion experiment to figure out what this specific worm does to the benthic habitat where it's abundant. But so to some extent, I'm interested in this specific species or organism, but to some extent, lugworms are a great model study organism to understand the impact of bioturbation on the seafloor and benthic communities. And here on this first slide, you get a little sense about my funding sources, about the institutes I was involved with, and you will hear more about it within the seminar. So looking back, this was really when I got really, well, even more intrigued by working on bioturbation, which was the first Nereis Park conference in Paris-le-Route, where I learned to know so many people, which was really inspiring for me. So this is the title slide of my short presentation I gave back then. And you see the title has not changed much, but I hope I will show some newer results also from this experiment within the seminar. And I really want to start thanking some very important collaborators, mentors along the way. Carsten Reise, who is now somewhat retired from the Alfred Wegener Institute in Germany. He was the section leader of the Wannsee station for a long time. He was in Germany, we call it my doctor father. He basically came up with the idea of excluding lugworms from larger areas. Luboš Polaretsky, who was at the Max Planck Institute for Marine Microbiology at the time. He's now at University of Utrecht. He really introduced me into planar optode oxygen imaging, and we have collaborated on many different aspects of the work I will present today, and it has always been a pleasure to work with him. And then lastly, here I want to really mention Sally Wooden and David Wethy, with whom I postdoc for 5 years in South Carolina. They introduced me to a lot of different ideas about benthic habitats, about cool sensors that I learned to use and I'm still using. So I'm really thankful for that, beside their mentorship throughout the way. And well, you are all familiar with this cartoon, I think, and I just want to emphasize that the lugworm is here. And actually, I think he's presented very well in blowing a lot of water into the surrounding sediment. And that's basically one of the main reasons why it's so important in these permeable sediments, driving biogeochemical processes and also affecting benthic communities. So for those who are not familiar with lugworms, they are abundant. Well, there are different species around the world. I'm going to talk a little bit about this later on. They live in J-shaped burrows. The J is lined with mucus. While the feeding pocket is kind of open to the surrounding sediment, they inhabit sandy sediment, so they do not really occur in high abundances when it's getting too muddy, because they need to pump water into the surrounding sediment, and that only works if the sediment is permeable. So they live in these muddy sands at about 20 to 30 centimeters deep down in the sediment. Obviously, juveniles are located shallower, but in principle, they do the same thing. They subduct sediment from the surface through this feeding, sand column. They subduct surface sediment from the surface down, where they ingest it and then produce these fecal piles at the sediment surface. Where they are abundant, you typically can dig down and you will find a lot of shells below feeding depth, because over time they simply get buried as they are not ingested and then expelled by the worm at the surface. When you dig into the sediment, you see this oxygenated pocket already showing you how intensively they oxidize the sediment surrounding the burrow. As I mentioned, there are mostly older studies that looked at the global distribution, at the biogeography of lugworms. There are, again, like many species. The circles here, this is from a paper from Wells from 1963. So Arenicola marina is really like a cold water species, mostly in Europe and in the northern parts of North America. But there are other species in the Pacific region. They belong to the genus Arenicola. Today, you will see, I will mostly talk about Arenicola marina, which is the northern Atlantic species. As I mentioned before, they subduct sediment, and you can see this nicely if you do these time-lapse imaging of worms in ant farms. You see some activity down here. You see how the sediment is subducted and then pooped here out at the sediment surface, eventually overflowing through the out of the ant farm here. The pumping occurs in the opposite direction, tail to head, and again, it's injected into the surrounding sediment, and they do this by these peristaltic waves that you can see if you have animals outside of their burrow. Here are some juvenile Arenicola pacifica, but the Arenicola does pretty much the same. So they use this lined burrow, which gives some resistance when they move their peristaltic waves over their body, and by this, they basically inject small, about 0.1, 0.2 milliliters of water, which each of these peristaltic movements into the sediment. With 30 lugworms per square meter, which is an abundance that you can frequently see in areas where these worms live, the impact on sediment reworking and water pumping can be quite high. So with 30 lugworms per square meter, you have about, well, the upper 15 centimeters reworked once per year, or if you just think about the upper centimeter, it's reworked 15 times a year. And at the same time, lugworms at these abundances pump about 3 liters of water into the sediment per each square meter. They alter, obviously, the surface topography with this pit and mounds that they create. And for a long time, also, people were interested in the lugworm burrow itself and what it does to other organisms. Specifically, Carsten Reise had looked a lot into the little organisms that basically benefit from what the lugworm is doing. So there's a whole range of small organisms, amphipods, little nematodes that reside in high abundances in specific regions of the lugworm burrow. Here, for example, you can see a little amphipod that apparently is very abundant in the area where the worm is down in its gallery, and very likely this amphipod benefits from the oxygen that is supplied by the lugworm. Beside these small-scale implications on other infaunal organisms, myofauna and smaller macrofauna, obviously there's an interest in figuring out what these worms do on a larger scale. And I guess most of you are familiar with this concept of ecosystem engineering. So one way to figure out what these animals are doing, or a very powerful way, is doing exclusion experiments. And obviously, I'm not the first one who has done this. Carsten Reise and many other people have actually done this on scales like a quarter of a square meter or maybe a square meter to figure out what happens if lugworms are not there. What is the impact on the sediment? What is the impact on the benthic community, on biogeochemical processes? So my work, really, much of my work is focused here in the northern part of the Wadden Sea, close to the island of Sylt. There's a small harbor called the Königshafen, and this is really where I did most of my work. However, well, that extended to other areas, as you will see throughout the seminar. So Carsten, I did actually also like small-scale exclusion experiment during my master thesis. And then Carsten eventually came to me and Nils, let's make something larger, because with these small plots, it's very likely that some impacts you will not be able to see because there's lateral transport of sediment. And so the edge effect of your experiment may actually be larger than the plots themselves. So he suggested to me, let's go bigger, maybe something like 20 by 20 meters. That's easy. We just take a backhoe. We dredge away the upper 10 centimeters of the sediment. We roll out a mesh. And then when we put the sediment on top again, we get these nice exclusion areas. So we can then look into the sediment and figure out what's happening. So that's what we did. This is me when I was a little younger, less gray. We went out with a backhoe over, well, I think like 4 or 5 weeks or so. We dredged away the upper 10 centimeters. I rolled out the mesh, making sure that everything is nicely attached below ground when we put the sediment on top again. And we ended up with these really nice exclusion plots. So you see one 20 by 20 meter plot here. In the background, you see another one. Overall, I established 6 of these plots. In parallel, we established control plots where we dredged the sediment in the same way, but we didn't insert a mesh. And as a third type of treatment, we had total control plots where we did basically nothing. But I continue to monitor what's going on these totally undisturbed areas. The first thing maybe I should mention that the exclusion worked very well. So after the lugworm exclusion plots were established, basically no adult lugworms were found at any time on these exclusion plots. On the control plots, so these are the ones that were dredged, and the ambient plots, which were undisturbed, there's some variability, and specifically after 2 years or so, we saw this significant decline in lugworm abundances, which was obviously a little sad because that minimized the contrast between the exclusion plots and the control plots. However, that was already towards the end of my PhD, and I will show you later on how this continued to develop over time. I was very excited when in 2002, the National Park did some airplane photographs of the Wadden Sea. And when we saw that these plots were visible on these airplane photos, remember this was before Google Earth, right? So Google Earth was launched in 2005 or so. So airplane at the time was really the only way of seeing something from some distance. So that was obviously very exciting when we could see these plots. And at this time, I was not really sure why you can see them. Partially, it has to do with the fine particles, but also it had something to do with algae. And I will, again, get back to it a little later. However, after some time, obviously, this satellite imagery got available. And here you see, for example, and I continue to look at whenever it was updated, I continue to go to Bing Maps and to Google Maps to see if we can see something. And sure enough, in 2019, Bing Maps actually had this nice footage here where the area was exposed at the time. That's obviously important to see these exclusion plots. And you see here, if you look carefully, exclusion 1, exclusion 3, 4, 5, 6, they were all visible from space. And just to introduce you a little bit more to this experiment, we had basically 6 of these exclusion sites, 3 of them in the low intertidal area. So there the water is gone maybe for 2 or 3 hours per low tide. And the high intertidal area where it's more like 6 hours, the time that they fall dry. And again, we have these exclusion, these control sites established in a similar way. So again, like just giving you an overview about the experimental design here. It's not only tidal range that differed, also the sediment characteristics are quite different in the low intertidal zone, where the grain size median is maybe about 200, 220 micrometers, while in the mid intertidal, it's really like a more coarse sand with a grain size median of about 340 micrometers. And the impact of lugworms was quite different in the low intertidal fine sand compared to the mid intertidal medium sand, typically much more pronounced in this low intertidal fine sand. And again, like in just like 1 year ago, there was again an update on Google Earth. And again, I was really excited. I'm still getting excited if I see this. So again, like these dark exclusion plots, this one is not so visible, but the other 6 were quite visible. And I saw this on Google Earth and remembered that, wait, in March, I was on Sylt. And whenever I have a chance to be on Sylt, sometimes or oftentimes not because of research, but just visiting friends. However, I always take the time to walk out and do my little tour around these plots. So I looked back and I remembered, oh yes, something was going on. And indeed, at the time when I was walking over the tidal flats, I saw this bloom of tuft-forming diatom, Berkelaya, which was very apparent when I was there. I had seen something like this during my PhD thesis, so I thought, well, that's nice, that things are happening again, so it was not a one-time observation. And I will talk about this algal bloom later on. Again, like whenever I'm there, I go out, just like having a dog doing your tour. I do my tour around the exclusion plots, and there were some other interesting things, I thought. During my PhD thesis, I did a lot of work together with other people. For example, here we did some work on whether or not seagrass would benefit from the absence of lugworms. And when we did this Zostera nolti transplants, we decided to cut out the mesh in this area because we wanted to avoid that the mesh at about 10 centimeter depth would basically cause an artifact because maybe the roots get better attached in this kind of setting. So we cut out a 1/4 square meter of the mesh. When you now go back, actually these little cutouts have turned into little inclusion plots. So the lugworms are clearly able to find those locations. In fact, oftentimes the juveniles settle on these exclusion plots. Eventually, as they grow, they move out. But in these areas where we cut out the mesh, they don't have to move out because they can dig as deep as they want. And so they are quite happy in this area. So I want to give a brief overview over the years. So I introduced you now a little bit to the experiment that was established in 2002. And over the years, initially, the idea was mostly looking at the benthic community and the sediment properties during my PhD thesis. I then learned to know Luboš Polaretsky and Dirk de Beer from the Max Planck Institute. As I mentioned, they introduced me into planar optode oxygen imaging. I then learned to know David and Sally at an ASLO meeting. They heard about my exclusion plots. At the time, they started to work with pore water pressure sensors, and they thought it would be a great opportunity for them to come to Sylt and apply those sensors. And I'm going to talk a little bit about this work in a moment. That turned out, well, eventually they asked me if I would like to come to South Carolina. And so during that time, we invented the RoboLux, which are basically, we used the pressure sensor data to program peristaltic pumps to pump water exactly or close to what the worm do, with the same kind of, inducing the same kind of pressure fields in the sediment. We then also started to use hyperspectral imaging to look at the sediment surface, and this allows you to get the distribution of chlorophyll A and thus microphytobenthos. So Luboš was involved in this, a PhD student at the time, Arjun Chenu was involved in this, And Sally and David, they also managed to get a camera for South Carolina. So we had access to these technologies and doing work on both sides in Germany and in South Carolina. Once I started working here at Stony Brook, Jingxi Zhu, as you may know, he has developed together with Bob Aller a range of different planar-optols. So I was really excited when we started to collaborate to not only image oxygen, but also pH and CO2 within the sediments. And my last visit here was really during my sabbatical 2 years ago, where together with Dirk de Beer and Basu, we looked at hydrogen, at reactive oxygen species in sediments and iron mobilization in feeding funnels. I will not talk about this today, but I just wanted to mention that in 2021, I had another opportunity to visit Sylt and do some research there. So going back to the experiment, some things that I continue to do whenever I have a chance are simple things. So, for example, just figuring out, is the mesh still there? And for example, how deep is it? So I just put a ruler at 30 locations on each plots, and I measure when I hit the mesh. And you can see that over time, the mesh seems to become a little bit deeper, maybe 1 or 2 centimeters deeper than when I established it. However, the depth is not, as you see with the standard deviation, it's not the same everywhere. So there are some places where the mesh actually surfaced. There's also ice scouring, more rarely now, but it still happens from time to time. So, however, the mesh, most of the mesh is still buried and still excludes lugworms. I also did not do it frequently, but at least when I have a chance, I look at lugworm abundances. You saw, I already showed you this initial decline, and you see now here that this decline was really apparent in this low intertidal region. In the high intertidal, the lugworm population is stable, and there are still like densities of 20 to 30 individuals per square meter. However, in the low intertidal, it really crashed, and we don't know really why that is. Somehow it has now established at a low density. So on average, on these exclusion plots in the low intertidal, maybe there are 10 individuals per square meter. In some areas, however, here around exclusion 2, you barely find lugworms on the control plots or surrounding the exclusion plots. A very important impact that I found early on and during my PhD thesis is that lugworms maintain permeable sediment characteristics. So you see here permeability measurements that I did during my PhD thesis and another set of measurements that I did in 2019. You first of all see this strong difference between the low intertidal and the high intertidal, largely related to the grain size that dominate at these different locations. But you also see the difference between the exclusion and the control sites in the low intertidal. That has changed a little bit. Maybe the permeability have a little bit increased over these 14 years, but the significant difference is still there despite low abundances of lugworms on the control side. So it seems like this exclusion early on, is still like maintained within the sediment. For some of you who are not familiar with permeability, it's basically how much pressure you have to apply to force a specific amount of water through a given area. In simple words, how easy it is for water to flow through sediment. So you can do easy experiment where you put different pressure heads over the sediment and then you measure the flow rate. And this is how you can calculate using Darcy's law, the permeability or the hydraulic conductivity of the sediment. So here, oh no, there's a fire alarm here. What do I do? Can we, I think I really need to move out. Maybe you hear the bell. So should I continue or?
Please go ahead.
All right. So sorry about that. So I was talking about permeability, the exclusion plot that turned out to be less permeable. And yes, permeability is a measure of how easy water can flow through a sediment. And however, it's very important, a way to visualize how important permeability can be is when you look at how oxygen is penetrating for a given pressure head. So this little movie shows you, as we apply pressure across the sediment column, and these are now sediments taken from a control plot and from an exclusion plot, you see how differently or how much deeper oxygen is penetrating into the sediment from above in a more permeable sediment than in this low permeable sediment that I got from the exclusion plots. You see some activity of worms, likely Nereids, in these cores, but more importantly, you see how much more efficient physical advection by waves and currents will percolate those more permeable sediments. So by lowering the permeability, the sediment really changes in terms of its character, in terms of lots of biogeochemical processes, given that oxygen is so important. And Markus Hüttler has obviously done a lot of work on these permeable sediments and how effectively they remineralize organic matter that can be pushed into the sediment by physical advection, then be degraded, and all the products of these mineralization mineralization processes are then flushed out of the sediment. So you have these permeable sediments with a low standing stock of organic matter, but it's not because they are not active, it's because there's a very rapid turnover of things. So after these few introductory slides, I want to give you a little bit more, a little overview about what I'm trying to cover today, to get a, to get a sense of why I think lugworms, or why lugworms seem to be very important in these systems. You can think about these physical effects, and I just started to talk about that lugworms maintain these permeable sediment characteristics. I want to talk a little bit about the hydraulic activities that are related to bioadvection, and how organisms like lugworms can also change the local permeability of the sediment by fracking. I want to talk a little bit about the redox oscillations that are associated with their irrigation activity. They are obviously biological effects that I try to summarize a little bit, that most of the time are sediment mediated, which means lugworms change the character of the sediment, and so and therefore, and as a consequence, other infaunal organisms respond to that in the way how they recruit or not recruit those areas. I want to also talk about some of the biogeochemical effects, and specifically the gardening of microphytobenthos by lugworms. Briefly, I want to talk about some geochemical effects that I have looked in over the last few years, specifically related to carbonate chemistry. And obviously, overall, I want to give you a sense of the ecological context and some of the implications of all this work. So I mentioned earlier that pore water pressure sensors are a really useful tool that David and Sally invented to study the activities of infaunal organisms. The idea is here, as animals move through sediments, pump water, this should produce little pressure gradients within the sediment. And if you have sensitive pressure, pore water pressure sensors, then you can detect those. And sure enough, that's what's happening. So Sally and David did a lot of work in studying different organisms. Here are some pressure data from lugworms. And depending on what the animal is doing, you get specific waveforms. So the big negative signals are the signals that you get when a worm is pooping out about 1 or 2 milliliter of sediment to the sediment surface. That creates a negative pressure within the sediment. Most of the time, however, you see these little oscillations, which are related to each of these peristaltic waves that go over a body of a lugworm. Every now and then you also see these more irregular patterns, or these larger pulses, positive and negative. And when you look at, if you have worms in an aquarium, you will figure out that this is associated with the animals burrowing through the sediment. So most of the time, these animals pressurize the sediment. There are some behaviors that relate to these negative pressures. And these pore water pressure sensors are really powerful. You can record data over long periods of time. You can figure out, for example, episodes when these animals are burrowing. These are these large signals. But even like these areas, just a random section here in this pressure record, which looks kind of boring, you will get these specific behaviors, and you can count them, you can get their frequency. So a great tool to study the response of organisms to environmental stimuli or environmental changes. If you think about pore water pressure and what it means, it's again useful to look at the sediment with imaging techniques, and a very simple technique that many of us have used in the past are inert tracers that you can inject into the sediment. You can do it with particles. These are the red ones here at the sediment surface, but you can also do it with liquid tracers like fluorescein. And when I start this little video, you will see down here a pressure record, blue recorded down here, white recorded up here. And as the movie starts, you will see the red line moving, and I encourage you to look at the bottom graph, figure out whenever there's a strong positive signal, and then, partially look at the upper image to see what the fluorescein is doing. So here are the injections at multiple locations, and you see how effectively this fluorescein is driven out. This is not done by lugworms. This is actually multi-species ant farms set up by some students in a course that I taught. And you can see that just over these 6 hours, basically this entire fluorescein is driven out by the pore water pressurization of this informal community. So Sally and David were really excited to visit me at the lugworm exclusion plots on Sylt, because one thing that is obviously interesting to know is how far do these pressure gradients, how far are they detectable? And so the exclusion plot were a great opportunity to do this, because you could we could apply them from the exclusion plot. There is no, well, let's say from close where a lugworm is, so close to a burrow, close to a pooping pile, and then a transect of pore water pressure sensors. So with the idea, if we are close to the animal, we will get a large signal, and the signal should then decay over distance, so smaller and smaller, and we can then assess how far the signal is propagating. And indeed, a big signal like defecation, we could see on multiple sensors here, which were 10 centimeters apart. And from this, we could then look at, okay, at which distance, what proportion of the pressure sensor is still detectable. And from this, David developed a model to figure out what the pore water turnover is. And we came up with a number that around a burrow with the radius around a burrow of about 12 centimeters, in this region, 10% of the pore water are replaced per day. However, there's obviously not one lugworm here. There are 30, 40 per square meter. So if you draw more of these circles, you quickly can see that many regions, if you have an overlap of spheres of influence, there are regions within the sediment where it may just take a day or two until the entire pore water is replaced. So after doing this work, looking back at the data that I collected early on, they made even more sense. So one thing that I did during my PhD thesis, I copied a pore water lens that Markus Hüttel once built to study pore water profiles in these kinds of sediments. So these are 6 syringes where you can draw pore water from 6 different depths. So I could go around on these plots and always collect a little bit of water, pool samples from larger areas, do this on all exclusion plots. And what is very apparent that how efficiently this pumping, these pore water gradients, the sphere of influence affects the profiles of pore water constituents, including here, for example, ammonia, phosphate, silicate, so major nutrients where you see on the exclusion plots, as you go down, there's an increase in concentrations of these nutrients, while at the control and ambient sites, experimental areas, you don't see this accumulation. You see basically this effective flushing of lugworms down to 20, 30 centimeter depth where they inject the water. You can also see, just looking here at the upper row, which are the nitrogen-related profiles, that it seems like there's a lot of nitrification occurring with all this oxygen being flushed into the sediment. So hardly any ammonia, hardly any nitrate and nitrite on the exclusion plots, but appreciable concentrations of on the control and ambient plots, suggesting that these worms really keep, help to nitrify a lot of this ammonium that is produced by organic matter mineralization. And you can see this even more strongly if I did profiles across the boundary of an exclusion plot to the surrounding sediment. So you really see the steep change of pore water conditions from the exclusion plot. So the mesh here at about 10 centimeter depth with a lens, I could push through the net and even get like pore water samples from below this mesh. And you can see, we could clearly see that like this high concentration in this example in silicate below that mesh, an area that was not bioturbated by lugworms. At the same time, we found in these lugworm exclusion plot an accumulation of fine particles. Again, this is directly related to the loss in permeability, which was largely driven by this incorporation of fine particles on these exclusion plots in these fine sand sediments. So fine sand here means these are the averages of the 3 exclusion plots in the low intertidal, where the sediment was already kind of low permeability, not very permeable, but it went even less permeable after the worms were excluded. So one of the outcomes of my thesis was then that these lugworms really maintain these permeable sandy sediments. And in the Wadden Sea, you typically have these transition from marsh to mud flats. As you move out, the flats eventually gets pretty sandy, and this is where the lugworms reside. So the lugworms really help to maintain these sandy conditions. And since they are so abundant throughout the Wadden Sea and elsewhere, they really play an important role in keeping these sediments permeable. One of the processes that I thought would be the main cause of this was later on actually nicely shown by Wendelboe et al., who did a flume experiment. And they hypothesized because these worms, they bring up a lot of material in their fecal mounds. And as these fecal mounds are resuspended, lots of these fine particles are washed away. And this is basically how the high permeability and low fine fraction in the sediment is established, and the animals keep the sediment open and permeable. Permeability is also interesting if you think about the animal itself, as it needs to pump water into the sediment. So in South Carolina, I did a lot of experiments where I tried to understand what that means for sediments. So here you see an aquarium. And when I start the video, you see how I raise the head tank to an inlet that is somewhere here at depth, and basically figuring out, okay, if I apply a pressure head on the sediment, what is the flow rate? The graph is shown here. It's very straightforward. It's a linear relationship between the pressure that I apply and the flow rate that I can measure at any given time, as you can see here. So I actually don't see much right now, but eventually things change. Eventually, well, we establish a quicksand column, as you see here in the video. But even before that, you see that the flow rate actually increased faster compared to what you predict from this linear relationship, indicating that something is already changing in the sediment. Somehow the permeability effectively increases. Eventually, then I produced this quicksand column, and this was a poorly mixed, poorly sorted sediment with coarse sand mixed with fine particles. You see all these fines have been blowing out in this quicksand column. And if I then apply the same pressure heads afterwards, you see that, well, the flow rates are much, much higher. The slope is more than 2-fold higher, meaning if you would be in organisms that pumps water, after you establish a quicksand column, the energy that is needed, the force that you have to apply is less than half, which obviously makes it much easier for the organisms to pump water into the sediment, for example, a lugworm. And in this context, it's interesting to think about what kind of pressures can be applied by organisms. Some older studies by Foster Smith, lots of work done in glass tubes, figuring out against which pressure head organisms can pump. You see here arenicolids can pump maybe with against a pressure head of about 20 centimeters of water. So you see here, this is pretty much on the scale of this little experiment that I did. So indicating that worms, lugworms at least, not Nereis obviously, but lugworms can apply those pressures where things are changing. Looking back at the pressure records from lugworms, again, you sometimes see these positive and negative pulses associated with burrowing. Well, it's not only a reflection of what the animals is doing. We think that the worms actually, it may have a purpose. So again, you can do experiment where you apply a specific pressure, a constant pressure, you measure the flow rate, or here in this case, you apply a flow rate and you measure the pressure. And so we did this first here now for a constant pressure, for a constant flow rate. And then we used the flow rate, but added these small pulses of water by simply having the peristaltic pumps doing these quick injections of water, mimicking basically what the lugworm is doing when it's burrowing. And well, you can see in the oxygen video what's happening during that. So this is the constant flow rate that we applied. And the second time is now the constant flow rate together with these pulses. And you can see really like the small flashes here. And you can also see how some of these plumes change their location at the sediment surface, really indicating that something is changing, that the worm is basically cracking these sediments, opening highways of pore water flow, with the important implication that once the sediment is cracked, you see that the energy or the pressure that we can measure for the same pumping rate is about 20–30% smaller than what it was before these pulses were applied. So again, a way for the organism to, well, having to invest less energy in pumping the same amount of water. We see these same kind of plumes with animals in low permeable sediments here on the right side, while in highly permeable sediments with lower oxygen demand, you can really see this massive blooms of oxygen surrounding these worms. So depending on what kind of sediment type you are studying, the impact may be very different. I want to focus on both of these for a little bit. I want to start with the first one, very permeable sediments, little oxygen consumption, where you really see this massive oxygenation of subsurface sediments. And one thing I did at some point were having this flume where I could control the flow velocity over the sediment surface. Over time, the worms build up this microtopography. So my motivation was here to study both the impact of bioadvection induced by these lugworms at depth, but also the interaction of the topography with the current going over the surface. And what you see here is actually, I think, quite impressive. If you think about these worms at depth, here there's hardly any flow, but as I increase the flow, you start to see how powerful is also this physical advection together with the topography changes induced by these worms. So I don't mean to say that bioadvection is everything. Physical advection obviously is very important too, and typically they occur at the same time on these in these systems. So we can then use this kind of information of oxygen at depth and not only look at the distribution of oxygen, but thinking about how dynamic the environment is. So this here is now an image that has accumulated a lot of oxygen images and shows you the switches between oxic and anoxic conditions over 24 hours. So you clearly see where the worms are. You have areas where there can be 20 or 25 oscillations per hour. So every few minutes, the conditions switch between oxic and anoxic conditions. Well, maybe more like 10 to 15 in the case of lugworms. So that obviously has important implications for all kinds of processes on, for example, aerobic and anaerobic processes. How do microbes deal with such oscillations? Oxygen is obviously only one of these parameters that are really interesting to study. So I'm excited that we now could also do imaging of pH and CO2, oftentimes in parallel. So these are experiments I did 3–4 years in the past on Zul together with Ian Dwyer. So here you see 4 ant farms. 3 of these had a lugworm. And you now see here well, ant farms that either have only a CO2 optode, only a pH optode, only an oxygen optode, or strips of each of these combined. And we could image here from one side, we image basically CO2 and pH. And from the other side, we imaged oxygen. So we get co-registered information on oxygen dynamic and, for instance, pH. This is what you can see here. So this is an ant farm. On the one side, we had an oxygen optode. On the other side, we had a pH optode with, well, CO2 strips. I don't show this data here, just to give you an idea about the spatial impact that a worm has on the pH profiles within the sediment, or the distribution of pH within the sediment. I think it's getting pretty clear where the worm is mostly injecting water. And you just as a comparison, so if we now look at the pH that developed over time in an aquarium without a lugworm, well, this is totally out of scale. So here I scaled the image so that it has the dynamic range that we saw in the lugworm aquarium. Without a lugworm, it would be way more acidic. So I really need to use a different scale here. And you see that much of the sediment has a pH of around 7 or so, while this is barely reached in the lugworm, in the aquarium with lugworms. And we have these co-registered data to really confirm that where the animal injects water, overlying water, which obviously has a much higher pH, this is the region where we see this, these low pH conditions compared to the surrounding sediment, and specifically compared to the sediment that is diffusion dominated. Over time, so this was just a short episode. So here I briefly want to show you the longer term consequences of this. So we started with aquaria with constructed sediment. We actually had a shell debris layer at the bottom, and we included this iron sulfide-rich sediment and topped it with the surface sediment, so constructed sediment. In the beginning, they look alike. The profiles look alike. So you see here an image, and on the right side, you see a profile, basically horizontally averaged values for each pixel line. In the case of the non-irrigated aquarium, it's about 100 pixels. And since we have a much larger area here, well, over about 1,000 pixels. So now I start the video, and you can see how quickly the pH conditions in the irrigated aquarium changes. The worm sometimes flips, changes orientation. So that's sometimes why you see the impact more on the left side or more on the right side. And this is now after 15 days. And you see the average profile nicely shows this depth of injection of the lugworm, where the water is injected, but overall the pH stays above 7.6 pretty much everywhere, while it's way below 7.6 if you don't have a lugworm in this aquarium. And you specifically see this very low pH, well, just a few centimeters below the sediment surface. You have these very, very low pH values, largely related probably to the reoxidation of reduced metabolites that diffuse upwards from the bottom. You also see this higher pH, which we think has to do with calcium carbonate being dissolved here at the bottom, where we have this calcium carbonate shell layer. So with worms, it's very nice to study worms and using worms. However, sometimes it's annoying because the worms will not do what you want to do. So over the years, we have done lots of work with irrigation mimics. In this case here, for example, again, combining information from pH, CO2, and oxygen optodes in these optocosms. In this experiment, we pumped water with different irrigation patterns. So 30 minutes of pumping, 30 minutes of resting, or 12 minutes pumping, 12 hours of pumping, 12 hours of resting. And we can compare it with continuous irrigation. So here's just an idea to show you how this looks like. So you can clearly see the aquaria that received this intermittent irrigation. And just now, some of the oxygen blobs disappeared because that's now the 12-hour resting period. So we can, in principle, nicely mimic and simulate whatever oscillation frequency we want to look at. And we can then further study things like oxygen consumption rates in these specific regions. So this here is now one of these oxygen pockets. You see high oxygen consumption rates at the outer sphere of this oxygenated pocket. Again, this may not be surprising. As organic matter gets burned, oxygen consumption rates will decline. Plus, at the outer sphere, the injected water is in contact with this water that has accumulated some of these reduced pore water constituents. So this is the graph that exactly shows that. Initially, with a 30-minute irrigation, we have relatively high oxygen consumption rate in the same square that I show here. They relatively quickly decline over time. With a 12-hour regime, obviously we have less than, we cannot estimate oxygen consumption rate that's frequently. They're a little bit higher than the 30-minute, but over time they actually are pretty close to each other. So in terms of oxygen consumption rate, there is not a huge difference between, well, frequent irrigation and, well, continuous irrigation, let's say. So it seems more like a matter of how much oxygenated water is injected over time. However, these experiments, you really introduce the same kind, or you establish the same kind of oxygenated pockets that we see with real worms. And so here again, just an example of these co-registered data during continuous irrigation, 12 hours, 30 minutes. And again, we can then plot, for example, the dynamic range of pH that we see in these regions within the sediment here with the gray bars over this experiment. In these pockets, pH is oscillating by, well, more than 0.5 pH units over each irrigation cycle, over these 12-hour. The dynamic range is a little smaller, or much smaller, obviously. If you irrigate more frequently, there's just not much time for protons to build up within the sediment. So with a 12-hour irrigation regime, we see these delta. This is a delta pH image that shows you that between the onset of irrigation, or, well, when irrigation stops towards the end of the resting period, you see the pH declines by about 0.6 pH units. And we can further investigate this in terms of what is the pH change during 30-minute resting, which is shown here, and compare it to the maximum oxygen expansion after the irrigation. So you see here how the maximum oxygen expansion is increasing over time. And you pretty much can see this high, very dynamic pH region at the outer sphere of this oxygenated pocket. So from this, we can then further study the dynamics and get rates of proton production. For example, here you see the pH and how it's declining over time after pumping, after we stop the pumping. The dynamics are obviously very different with a 30-minute regime, but you can calculate for both of these experimental settings what the pH change per time is. So you get kind of the rates of pH change. We also have the CO2 data. So if we combine this information, we can even think about the carbonate conditions, the carbonate system within the sediment. And as you can see here, it changes dramatically from, well, the end of an irrigation event towards the end of a resting event, from supersaturated conditions down to undersaturated conditions. And that holds true for the oxygenated pocket. That holds also true for the bulk sediment. So here you see the typical pH ranges that we measured in the bulk sediment. And if you compare it to the non-irrigated sediment, again, the pH remains much higher, but also the aragonite saturation remains typically at 1 or even above compared to this very undersaturated conditions within the non-irrigated sediment. That obviously may have implications for the fate of calcium carbonate, for example, produced by bivalves within the sediment. You would expect that these calcium carbonate shells dissolve relatively quickly in the sediment that are very undersaturated. So another thing that we try to understand a little bit better, again, ant farms with and without lugworms. This is now information not gathered from Planoroptals, but from extracting pore water, looking at the pH without a lugworm, looking at the pH with a lugworm, measuring alkalinity. And from these values, we then can calculate aragonite saturation. Again, with a lugworm, aragonite saturation oftentimes remains above 1. However, you have these very undersaturated conditions at very shallow depth, but in this, well, sediment without a lugworm. So one of the questions I was asking myself at some point, we have this beautiful shell layer in areas where lugworms are abundant or have been abundant. So after 20 years, I thought, well, it could actually be a great opportunity to figure out if the shell layer is still there, which has now been buried for 20 years below the mesh. And I give you a second to decide what you think happened. If we would be in a room, it would be nicer. I could see hands. Well, the outcome was it's still there. There are some indication that the amount, so basically what we did, we cored through the mesh, through the shell debris layer to an area where we don't see much shells at all. And then we sift out the larger particles, the calcium carbonate, the shell material, we weight it. If you do a matched pair test, it is significant. However, you see that the amount of calcium carbonate larger than 1 millimeter in grams per square meter is still like comparable. Slight indications that some of it has been lost over time, but it's not gone. It has not melted away over these 20 years. Another observation that we did is that there was no shell accumulation above the mesh. So that's interesting as well, because that means that any bivalves, any shells that have been produced during that period of time has not been accumulated above the mesh, but likely got dissolved due to these corrosive conditions. So lugworms likely not only produce the shell layer because these particles are subducted, but also because the sediment has the characteristics that the calcium carbonate shells are less likely to be dissolved. A few more things about advection and how important it can be. A recent work that we did here at Stony Brook with Bob Aller and Laura Wehrmann and Darcy Swenson-Perger, and there are papers just now submitted or already published. We looked at iron fluxes, again, using irrigation mimics in these benthic chambers, injecting water. We used little iron traps, and if you want to learn more about those, please check out the Bob's paper on that. And I don't want to show data here, but I want to show how different iron concentrations were in the pool water in these non-irrigated cores, which you can see here from the yellow color, and what we trapped in these iron traps, which is shown up here, where you see a lot of iron fluxing out. And this was a sustained flux. So it was not like this iron was simply flushed out. We actually, over many days, we continue to see this a lot of iron being mobilized in these irrigated columns and then being fluxed out and eventually trapped in our iron traps. I briefly want to talk about the right side here, which is related to these anoxic plumes that we always see when we have these sediments that are low in terms of their permeability. These plumes, they are dynamic. As you can see here, this is here on the right is a profile over time through one of those plumes. From the pore water pressure record, we know that these dynamics are directly related to the activities of the animals. So blue, for example, is always when we see a pressure below 0, so a negative pressure. And that's exactly when these plumes collapse, or like even it relates to the opposite, that oxygenated water is drawn through the sediment surface to a few millimeter depth. You can also see this if you look at a profile perpendicular to the parallel to the sediment surface. You see that there are areas where these plumes are more likely to emerge when the animal is pumping. You see a lot of heterogeneity on the sediment surface, and that this heterogeneity also changes over time as the animal is moving and potentially opening new cracks through the sediment. We can also see, and one idea here was, so if there's lots of anoxic water coming out, this anoxic water probably is also rich in nutrients, and therefore may be very beneficial for microphytobenthos at the sediment surface. Here we have a little hotspot of microphytobenthos. We put an oxygen microsensor there. And indeed, if we have a large negative pressure signal, in this case by an arenicolate, this is exactly the time when oxygen concentration in this location actually increased, because the oxygen-anoxic boundary was actually moving downwards during this during this negative pressure signal. So we used hyperspectral imaging, and that was work. Arjun did his PhD thesis on this. We did, for example, experiments in South Carolina where we had these buckets, again, irrigated with these peristaltic pumps with Robolax. Some of these buckets were irrigated, others were not irrigated. We scanned the sediment surface. Clearly some microphytobenthic growth and lots of oxygen production during the daytime. These are now surface images of these buckets, and you see clearly this vertical migration of microphytobenthos during day and night. So higher concentrations during the day, low concentrations during the night. But over time, you see also that these irrigated buckets, they show a much more productive system in terms of microphytobenthos productivity. So after a couple of days, you see this clear difference in microphytobenthos in these irrigated buckets compared to the non-irrigated buckets, confirming the hypothesis that these worms really, by what they are doing, they fertilize the microphytobenthos at the surface, which is interesting because this is exactly what they feed on. In the exclusion plots, on the other hand, in this field experiment, the outcome was opposite. I always found higher microphytobenthos, higher chlorophyll A in surface sediment without the worms. But this is again an example of how you can fool by some experiments. In the field, lugworms obviously feeding at the same time. So in our experiment, we basically decoupled the bioadvective fertilization of microphytobenthos from the grazing, from the predation of microphytobenthos by the worms. So something that you can only see if you decouple those two processes. So it's again a good example of a relatively low standing stock, in this case chlorophyll, but it's not because these systems are not productive, it's just the opposite. They are very productive, but the standing stock remains low because of this high rates of feeding by lugworms. And you can certainly see this if you just scan in the field, these lugworm populated areas where you see this blue lugworm poop, very cold because all the chlorophyll, all the microphytobenthos has been digested, but you also see these hot spots. Lastly, I briefly want to go through some of the impacts on the community. So early on, we saw this invasion of adult Nereids. So I really had Nereis Park here on the lugworm exclusion plots. Other worms like Scoloplus, they benefited from lugworms being present, so they were not very abundant on the exclusion sites. Over time, I had the opportunity to, close to the shore, you could go there every day. So there are simply things that I obviously quantified, but I just want to show it here, like the settlement of juvenile arenicolates that really like these exclusion plots, sometimes 2,000 per square meter or even more. We had these Lanice settlement, tube-building worms really benefited from lugworms being absent. Sometimes there were these algal blooms. I showed you a picture earlier on. Sometimes it was just opposite, clear microphytobenthic blooms in the presence of lugworms and not so much on the exclusion side. Oftentimes these things are interrelated. So some things you could repeatedly see on these exclusion sites where tube-building worms benefited from the absence of lugworms. And actually these tubes, they were the anchor for algae to grow. And all these above ground structures then attracted juvenile bivalves that tend to float around with the currents and then get attached to those subsurface structures, and in turn to be much more abundant on the exclusion plots. I want to end with some larger perspective, and Sally and David have just last year published this paper where they looked at future forecasts for Arenicola marina distributions with continued global warming. I encourage you to look at this paper. Basically, they modeled future Arenicola marina distribution, given that temperatures of 25 degrees at burrow depth can actually be lethal for them. So then they run the model with future climate conditions and the distribution of arenicolates, showing that these worms may actually disappear from some areas around Spain, potentially into France. And so with that, the question then is obviously if there's a functional equivalent to lugworms that would take over. In the case of the European coast, that's likely not the case. And I hope I try to show you in this seminar that this will really have strong impacts on the ecosystem. Worms have been there for a long time. This is a very nice picture that Gabriela Mangano took in northern Spain. Lugworm poop that looked like freshly defecated at the surface. However, this is 150 million years old. So they have been out there for a long time. I want to end with, hopefully I convinced you that lugworms fundamentally alter the sediment characteristics. They maintain the permeability of the sediment, and it seems like the exclusion plots have kind of established a new steady state. So the permeability has not dropped further. It's now pretty much as it was like 14 years ago. These habitats are characterized by low standing stock and high turnover of fluxes of a lot of constituents, including organic carbon, mineralization products in the pore water, chlorophyll, iron. So don't be fooled if you don't find something that is not very high concentrated at a location. It may just mean that that there's a rapid turnover of things. With respect to calcium carbonate, it seems to be the opposite, with lugworm tidal flats having a high standing stock of this calcium carbonate being preserved and then accumulating at depth. And the response of benthic community, I would call it, there are lots of contingencies, so you cannot really predict things, but the likelihood that specific things happen is higher on these, without the lugworm, for example, that tube builders take over, that provide anchor and so on. I want to mention that these exclusion plots still exist, and they will probably continue to exist for a little longer or for much longer. So maybe in 10 years, I can give another seminar. If you want to use them, just reach out to me or the people on Zult if they're interesting for your work, maybe. There are lots of people I need to thank, but I already thanked them before. So I want to end with thanking you for being here and the Nereis Park seminar series, the organizers of this. And as Frank mentioned, I want to propose this year. Happy to take questions. And I want to see some faces.
Yeah, you want to see the faces. Okay, so let's start the cameras.
Yes.
Thanks, Nils,
nice.
for this very